Hello, my name is Christy Renouche, and I will be presenting cytoskeleton movement. I'm from group 6, and I will start for question 1 and 2. Okay, so the article that we mostly focus on is Kinesin 8 specific loop 2 controls the dual activities of the motor domain according to tubular protofilament shape. This article was found under this site in which we cited it. Okay, first question that we will answer is, what was the main focus of the study? Meaning, what question were scientists trying to answer? Well, there's two. Well, we put in that investigated the role of a specific region called loop 2 in the kinesin 8 motor protein, as well as how it controls the motor domains, dual activities in response to the shape of tubulin protofilaments. The scientists were trying to answer the question as to how kinesin 8 motor proteins function in the context of intracellular transport, how they respond to variation in shape structure of microtubules composed of tubulin protofilaments. So basically, to explain the data and figure as shown in here, it was used to compare the ATPase activity of different kinesin motor domains in presence of the free tubulin curved formation. We tested 3 kinesin motor domains, in which 3 of them, the first one is CA-KIP3MD, also known as Cyanorhabditis elegans kinesin 8 motor domain. The second one is CA-KIP3-MD and L2-HSKHC, also known as CA-KIP3 motor domain with insertion of the loop 2 from the human kinesin 1 motor domain, and HSKHC MDCL2-CA-KIP3, also known as the human kinesin 1 motor domain with an insertion of loop 2 from CA-KIP3. Okay, to interpret the data explaining what we are seeing on this figure and what does this mean. So we are going to describe what is happening on these 4 figures on graph A, B, C, D, and E. So we are able to see that graphs A, B, and C, the x-axis for A, we are able to see that the ring is measured on their micromolars. As the y-axis, we are able to see the ATPase rate. So to explain for what's going on in graph A, in CA-KIP3, MD, and L2, HSKHC, the loop 2 region from CA-KIP3 is inserted to motor domain of HSKHC. It shows that CA-KIP3, MD, and L2-HKHC had no ATPase activity in response to the free tubulin. It does not respond well to the curved tubulin shape, as we are able to see on the gray area, as compared to CA-KIP3, MDN, which is the yellow dotted area, which there is a higher curve. As for in graph B, we're able to see that x-axis is under tubulin, under micromolars. As for the y-axis is the ATPase rate. We're able to see the difference of curves in CA-KIP3 MDN. It represents the motor domain of CA-KIP3 without loop 2 insertion. It shows the significance ATPase activity in response to free tubulin, responds well to the curved tubulin shape, as we are able to see on the yellow section of the graph. Compare the CA-KIP3 MDNL to HSKHC as it's stable. On graph C, we're able to see that the tubulin is measured under micromolars again, as well as the ATPase rate. On graph C, we're able to see in HSKHC, MDCL2, CA-KIP3. Loop 2 region from CA-KIP3 inserted to motor domain of kinesin 1. There is a significant increase in ATPase activity in response to the free tubulin, as we are able to see on the blue linear graph. Therefore, the insertion of CA-KIP3 loop 2 into kinesin 1 responds well to curved tubular shape. On figure 5D, this is just a graphic result of a summary comparing the ATPase activity and how they each respond on the free tubulin. The techniques that were used on this article is X-ray crystallography to visualize microtubules on bound ADP state of CA-KIP3. Second one is cryo-electron microscopy, a 3D structure of the microtubule bound CA-KIP3 also. It is used to see the structure of CA-KIP3. The last one is MDC bound to the dolostane stabilized curved tubulin rings. As we're able to see in the graph, we are able to see the curved tubulin profilament as well as on the orange kinesin 8. On this teal color is B tubulin, and alpha tubulin is this blue sky color. Okay. On question 2, what conclusions can you draw from these experiments? So the conclusions that we were able to see based on the experiments is that buffer and tubulin length of the tubule increase according to time. Microtubules have two critical concentrations. A lower critical concentration where individual dynamic, unstable microtubules can grow transcendently, and an upper critical concentration where populations of such filaments grow persistently. In this case, length increase shows an upper critical concentration, where the length of the tubule increases persistently. That is, there shows a steady increase of stability increase. When it is replaced by a buffer alone, at first there was a transient growth and gradually decrease in length observed with time, as we are able to see on graph A. What is the role of kin 1? We're able to see it on graph B. In this experiment, the tubule length increased along with time, as seen here. Kinesin increased the length of the tubule in upper critical concentration, which aids persistent growth, shows first and then followed by a transient growth with lower critical concentration. Kin 1 moves across microtubules by hydrolyzing one molecule of ATP at each step in a single direction to produce a walking motion. Kinesin motion is thought to be produced by either a hand-over-hand mechanism, where the globular heads altered and step across the microtubule, or by an inchworm mechanism in which one globular head leaves and the other catches up. Okay, what conclusions can you draw from these experiments? What is the significance of the nucleotide present in kin 1 activity? Okay, so we're able to see on this picture, the heavier particles will be found in the palate, while lighter particles will be found in the supernatant. In the CTLR ATP lane, the band present confirms the presence of tubulin in the taxol stabilized microtubules. In the lane showing result that added ATP and KHC, this band indicates no change in the tubulin present in this taxol stabilizing microtubules. Comparison the last two columns, ATP kin 1, AMPP, and NP kin 1, indicates the ATP hydrolysis causes shortening of the taxol stabilized microtubule caused by depolymerization. The last question, would you expect ATP to be present in these experiments depicted in part B of the figure? Why or why not? Well, we answered yes because ATP hydrolysis is required in order to facilitate depolymerization. And that would be all. Thank you.